Every successful pharmaceutical lyophilization cycle follows the same three-stage process. Whether you're manufacturing a monoclonal antibody, a vaccine, a peptide, or a diagnostic reagent, every product moves through the same sequence: freezing, primary drying, and secondary drying. Each stage has a different objective, but together they transform a liquid formulation into a stable pharmaceutical product that can be stored for months or even years. Understanding these three stages provides the foundation for everything else in pharmaceutical freeze-drying. Although we often describe these as separate stages, lyophilization is one continuous process. The product begins as a liquid formulation inside a pharmaceutical vial. During freezing, the water solidifies into an interconnected network of ice crystals. During primary drying, that frozen water is removed directly as water vapor, leaving behind a porous structure. Finally, secondary drying removes the small amount of moisture that remains attached to the internal surfaces of the product. Each stage depends on the success of the previous one, making the entire process carefully connected from beginning to end. The first stage is freezing. At the start of the cycle, the formulation is a uniform liquid containing water and dissolved pharmaceutical ingredients. As the temperature decreases, the first ice crystals begin to form. This marks the beginning of solidification. These crystals continue growing until the entire product becomes frozen. As ice forms, the remaining unfrozen solution becomes increasingly concentrated with dissolved components. The arrangement of these ice crystals is extremely important. When the ice is removed during the next stage, the spaces it occupied become the pore network of the final lyophilized cake. In other words, the frozen structure created during this stage determines how the product will dry later. Once freezing is complete, the product contains a continuous network of solid ice. The next objective is simple in principle, but technically challenging. Remove the ice without allowing it to melt. This is achieved during primary drying. Primary drying is the longest stage of the lyophilization cycle. During this phase, the chamber pressure is reduced, while heat is carefully supplied through the freeze-dryer shelves. Together, these conditions allow the ice to undergo sublimation, changing directly from a solid into water vapor without passing through the liquid phase. As sublimation begins, water vapor travels through the product and leaves the vial. Gradually, a dry, porous layer forms above the remaining frozen region. The boundary between the dry layer and the frozen layer slowly moves downward as more ice is removed. Eventually, all of the frozen water has been removed, leaving behind a highly porous solid structure. This porous network is one of the defining characteristics of a successfully lyophilized pharmaceutical product. Primary drying must be carefully controlled. If too little heat is supplied, drying becomes unnecessarily slow. If too much heat is applied, the product temperature may rise above its critical limit. When this happens, the delicate, porous structure can begin to collapse, reducing product quality and making the remainder of the drying process more difficult. For this reason, shelf temperature, chamber pressure, and product temperature must all be carefully balanced throughout primary drying. Even after all visible ice has been removed, the product is not completely dry. A small amount of water remains attached to the internal surfaces of the porous structure. During secondary drying, the shelf temperature is gradually increased while the chamber remains under vacuum. This additional energy removes the remaining bound moisture from the product. The objective is to achieve the residual moisture level required for long-term stability without damaging the pharmaceutical formulation. By the end of secondary drying, the product has reached its final lyophilized state. In this video, we've introduced the 3 stages of pharmaceutical lyophilization. In the next episode, We'll take a closer look at the first stage, freezing. We'll explore how ice nucleation occurs, how ice crystals grow, and why freezing is one of the most important factors influencing the success of the entire lyophilization cycle. Thank you for watching Lyophilization Core. If you found